Okay, so this course is going to be running the 13th through the 17th, Monday through Friday, mornings and afternoons. What we're going to be doing is something that is a little bit terrifying, which is attempting to teach Introduction to Computational Neuroscience using a tool that has only very recently been developed. So only in the last couple of years have we been actually developing Neuroblox. We have a beta version that is now being released, as of this week. And so you are actually the first users who are not the people who developed the software. That is kind of exciting, but also runs the risk that there may be bugs that we hadn't anticipated because you will use the software in ways that we hadn't anticipated. That's all a good thing. And so rather than fighting it, I suggest we embrace it. There is, within the Slack in general, bookmarked a report a bug. And so there's a difference between I'm trying to do something and I don't know how to do it versus I know how to do it and it's not working. If it's the latter, then please report the bug so that we can try to rectify it as soon as possible. So this is our team. As you can see, it's pretty interdisciplinary. It's also across multiple institutions. MIT is obviously very well represented here. Members of this team are going to be, some of them are physically here, some of them are monitoring channels remotely, but this gives you sort of an idea of who the main individuals are so that when I credit them during the talk, you kind of understand who they are, and these are also gives you a sense of who would be answering your questions. So if we're going to talk about intro to computational modeling, okay, so what is computational modeling? Modeling means different things to different people. If we're thinking about a material model, so the father of cybernetics had a very memorable statement: the best material model of a cat is another or preferably the same cat. And I mean, what does that mean? It means that if you want to characterize all of the features of a particular system, your model must be essentially have a one-to-one mapping between the features you're trying to describe and the components of the model. But Lemma's point here, Lemma is the cat, our cat. Her point is that you run the risk of overfitting depending on what the aims of the model are. So if your aim is to answer every possible question, then you need to essentially reproduce the system completely. But in most cases, we make strategic choices about what we're trying to accomplish with the model. And so Lemma says a computational model's level of resolution is a critical design choice that needs to match the system's goals. I agree with her. She's a smart cat. So different computational neuroscience software platforms exist. Some of them are very well developed and have accomplished a tremendous amount. Neuroblox distinguishes itself by its specific set of goals, which is really optimized for biomedical engineering applications. So what we're primarily interested in is mechanistic integration of disorder and treatment relevant targets. And that motivation is informing all the design decisions that we made in terms of the architecture of the software, what we leave in, what we leave out, what types of solvers we use. All sorts of technical and also conceptual decisions are based on that. So if we think about where our data come from, so they could come all the way from the bottom patch clamp all the way to the clinical signs and symptoms that we're trying to explain, so psychiatric and neurological disorders. And so you might be able to ask, all right, what is causing dementia? What is the time course of dementia? Who is at risk for dementia? What will be the progression of that disorder? Depression, addiction. Many of these are disorders that are not simply binary. They have trajectories. They might be cyclical, like bipolar disorder, they might be degenerative. They might have an onset, for example, with psychotic disorders that starts in men at one age and in women at another, and you'd want to understand why. And so what we want to do is to create a model that is capable of addressing those sorts of questions. In order to address those sorts of questions, we're going to have to ask, in terms of the modeling level, what are the targets potentially at all scales that might give rise to these emergent effects? So that means that we're going to need to be able to include components within our model that have something to do with ion channels, receptors, neurons, neural masses, brain regions, all the way up to the circuits that we associate with the macroscopic neuroimaging world. Ultimately, we output to clinical signs and symptoms, everything from behavioral outputs in terms of how someone might perform on a task, but also mood, for example, if we want to chart bipolar disorder. So what this means then is that if the mechanistic targets are at all of these scales, the treatments also might be operating at many different scales. So we want to be able to, within our model, implement different pharmaceutical treatments and ask what would be the impact of, for example, in this case, giving L-DOPA. We might want to ask about metabolism in the context of brain aging and Alzheimer's disease, where in general the field believes that metabolism has a really critical role to play. But what is the pathway by which this metabolic dysfunction actually then affects brain function, and specifically why some brain functions rather than others? And then finally, we might want to be able to ask of the model, tell us something about what happens when we stimulate the system in various ways. So if we administer deep brain stimulation or transcranial magnetic stimulation, we're influencing the system in some particular way. And bear in mind that the direction of influence is not just upward, there's also feedback effects that are working downwards as well. So we need to consider the causality from the bottom layer all the way to the top layer, but then also how elements of the top layer feed back down to the bottom layer. The final point I want to mention is that if, in fact, what we want to get out of this is something like homeostatic regulation or behavior on a behavioral task, then our outputs have to, in some sense, they have to be able to do a task. The model needs to be able to do a task. It needs to not just say this person will do it well, this person will not do it well. We want the model to actually elicit the task behavior that a human would perform. So that imposes all sorts of constraints, but it's good and actually I would say critical to have those ends in mind before you start. I should also mention that a lot of the models that you're going to be doing don't take advantage of all the capabilities we're developing, because as an introduction to computational neuroscience, what we're trying to do is to show you how the tool can be used with existing models, which are canonical and very important within the field, and then we'll give you an example of how it can be applied to the sorts of models that we're describing here. So in the world of neuroimaging, which is where I come from, there are terms that get tossed around a lot: networks, circuits, computational model, and that's true. I mean, these are real entities. These are important areas of research that have yielded really important insights, but different fields have different ways of understanding the same words. And so for the sake of clarity, I want to be kind of clear about what we mean by these things in this context. So in the context of neuroimaging, when we say networks, and even sometimes circuits, what is typically meant is something that looks like this, where we have a seed region, and then we're asking what other areas of the brain are co-activated with that seed region. So what we're really doing is taking a time series from one region and asking for the time series that are obtained from every other voxel in the brain, or every other region in the brain, depending on how you parcellate your brain, the areas that are associated with that first region that are somehow connected or communicating or signaling are described in that way based on correlation coefficients. You can also expand this to a general linear model where you have a design matrix and you ask in response to some particular experimental input what areas are activated in response to that input. Differentially, let's say, between one condition versus another, between one population versus another, but these are all statistical tests. When we talk about computational modeling or computational neuroscience within the neuroimaging world, what is often meant is really something very, very different than how basic neuroscientists would think of computational neuroscience. What's happening is that there's an emphasis really on developing tools that are able to make better use of the time series that are obtained from these voxels. So the sorts of computational tools that have been developed in neuroimaging, by and large, there are exceptions, really they hinge around two different concepts. One is improving reliability and reproducibility of connectivity, which, as we said, are correlations. Coactivation patterns, which are described through the general linear model, or feature extraction, often through independent components analysis, using meta-analyses across data sets and modalities. So given what has sometimes been described as a reproducibility crisis within the fMRI world, there's a very strong effort to see, okay, if we say these networks exist for one data set, do they also exist for other data sets? If we say they exist for fMRI, can we elicit them also from MEG? So the idea here is really to use computational power to enhance this reproducibility and reliability. The second context is using some methods for machine learning to decode functions and disorders from time series. And in this case, what's really happening is that people are identifying signatures, brain signatures, for this disorder versus that disorder, or for this function versus that function. And here, the advantage is that decoding actually can be done not just on time series, but on lots of different types of data. So you can use multimodal data and use structural information, functional information, all sorts of information together to provide these sorts of diagnostic tools. But for the purposes of what I've said in terms of our aims, those two approaches are not what we mean by computational neuroscience. So for one thing, what we need is to be able to simulate the impact of one drug or device versus another, to tune parameters and ask how the system will change, to ask how a patient who has undergone deep brain stimulation is likely to respond to tuning the parameters this way versus that way. All of this requires simulation. This requires an element of time. So it's not just asking, are you in this category or this category? It's asking, how does the system evolve over time? So both of these methods that I've described are discriminative. They are not, you can't simulate with them. You can describe symptoms, you can describe disorders, you can describe functions, but what you can't do is simulate how they will evolve over time. In terms of interpretability, machine learning has its strengths precisely because it's high dimensional in this context. There are types of machine learning, of course, that are generative, but that's not what's being generally applied to neuroimaging data. But because it's high dimensional, the flip side to that is that it's not interpretable. So you can't take those signatures elicited by machine learning from neuroimaging data and then reverse engineer what actually the brain is doing. Likewise, using multiple pairwise correlations to infer circuit architecture is not very reliable when you're looking at networks. So for one thing, circuits include feedback, and correlations fall apart when you're looking at systems that include feedback. The second part is that in a network, you have pairwise correlations, which if you add them, so if you have a particular network with nodes and you ask, what's the correlation between this node and this node, and then this node and this node, and this node and this node, the error estimates for each edge multiply. And as a result, when you ask what is the error of a single connection, the error for a single connection might be totally manageable. But once you get to a network with many edges, you have this exponential amplification of error. So it can very quickly get out of control. And actually, if you start with an R value, so Pearson correlation of 0.45, which is quite strong within neuroimaging, you'd be super happy if you got such a correlation. But that's only explaining 20% of the variance, which means 80% of the variance is not being explained. Now, if you have 3 steps, you have 0.2 cubed, which means that you're not explaining over 99% of the variance. That's not a very good model. So for that reason, it's not a great way to identify the structure of circuits. The other problem is that even if you were doing a great job of absolutely identifying the structure of the circuit with your networks, the other issue is that just talking about who's connected to whom is a very sparse amount of information about the structure of your model. And so as we'll see, there are a lot of functions, really critical functions within the brain that require a lot more information than who is talking to whom. So if that's not what we're using, if we're not going to be just relying on connectivity understood as correlation coefficients or general linear models, then what we'll be using is something called dynamical systems, which are based on ODE systems. So the advantage of ODE systems is that they address a lot of the concerns that I just raised. You can talk about the error of the system as a whole in a way that you can't with a series of correlations. You can model explicitly feedback, and also you can simulate. And so here I take as a really simple example glucose-insulin regulation. So this is something that probably all of you know about. You know, you eat a cookie, your glucose levels go up, but glucose levels being too high is quite dangerous, and so your endocrine system has a feedback loop that allows you to release insulin. Insulin then lowers your glucose levels. If your glucose levels go too low, then you release glucagon, and the glucose levels go up. So here you have a homeostatic regulatory system that maintains glucose within appropriate thresholds. And so what you can see here is if we give a glucose bolus, we can simulate how glucose and insulin will respond. So glucose will respond first, insulin will respond shortly thereafter, and then glucose will go down, and then insulin will go down. You can see that one of the nice features of this kind of model is that if we say that the circuit is dysregulated, we can be kind of precise about how it gets dysregulated. And it turns out that different ways of dysregulating the same circuit will lead to very different phenotypes. So in type 1 diabetes, unmedicated type 1 diabetics tend to be very, very thin because insulin is responsible for putting on body fat and storing it. Type 2 diabetics who are unmedicated will tend to be overweight because they have too much insulin and they're storing the glucose rather than consuming it. So if you were to just look at individuals with type 1 and type 2 diabetes and just look at those phenotypes, you wouldn't necessarily understand that those two disorders, which look completely different from a clinical perspective, actually represent different types of dysregulation of the very same circuit. So that is actually kind of provocative when we think about psychiatric disorders, because psychiatric disorders tend to implicate many of the same circuits, in spite of the fact that they can look quite different. And so this provides a kind of a way of thinking for how that might occur. Importantly also, I've just done a hypothetical experiment here. I've given glucose. I've seen how there's a response over time. But what's very nice about being able to simulate is I can ask, all right, if we chronically do this, how will the system change over time? So we can ask about longer-term trajectories. So when we do experiments in the lab, whether we're doing it in the scanner or we're doing it with electrophysiology, we have a very small time window in which we can conduct our experiments. But clinically, disorders, diseases, they evolve sometimes over decades. And so understanding how that process occurs is something that we can do with simulations. So one example here is with type 2 diabetes. If there's a chronically elevated demand for insulin, beta cells hit their carrying capacity. After a certain point, the beta cells, they're fatigued. They can't keep up with the demand. And as a result, type 2 diabetics can also become type 1-like diabetics. Again, this provides a way of thinking about certain types of psychiatric disorders that seem to flip states. So for example, from mania to depression, all sorts of states from anxiety to depression. There are all sorts of trajectory changes that occur over much longer periods of time than you can measure in a laboratory that you'd want to be able to explain with your computational models. And note that when I say carrying capacity, in the context of neuroscience, this is a very real concern because a lot of these dysregulations are putting a tremendous amount of excess stress on the system. And so if you are in a state like mania, where you're rapidly depleting dopamine, then what you're going to do is to put a lot of stress on precursors for dopamine, like tyrosine, for example, where acetylcholine has a precursor choline. And what can happen is that as you hit that carrying capacity, the system can collapse. So this is a really important dynamic, overshoot and collapse, that you'll want to be keeping in mind for the models. And finally, I just want to really bring home again the idea that what I've just described, this story of being able to regulate between glucose and insulin and to be able to dysregulate in more than one way, none of this can be captured through just asking who is connected to whom. So if we want to ask, all right, well, I said it's not just about excitatory and inhibitory responses, we might need more structure. So here's an example of this kind of modeling applied to the neuroimaging scale. And we can look at this prefrontal limbic circuit. Again, there are many similarities with what we've just seen between the glucose-insulin regulation. One of them is that we have not one feedback loop, but multiple feedback loops that are all interacting with one another. And that's one reason why you need to simulate the results, because these are complex enough that you can't just imagine, well, if I were to do this, this would happen. If I were to do that, this would happen. These are feedback loops that are operating at multiple scales. So at the bottom scale affecting the top scale, but also at each scale, you have multiple feedback loops interacting with each other. But I also want to point out that one feature of this particular model is the importance of gating mechanisms. So there are several gating mechanisms here. One is the thalamus, which is important for sensory gating, but also through the IFG in terms of other types of gating, determining whether there's object recognition or not, and that affects the feedback through the ventral stream. So having these gating mechanisms, thresholds, and also timing issues, the question of whether some signals are faster or slower than others is going to affect the dynamics as well. This is something, again, that OD systems can provide. So like type 1 and type 2 diabetics, depending on how the system dysregulates, determines the output in terms of behavior. And so we were looking at a largish sample of 256 individuals, all the way from clinically anxious to clinically reckless and everything in between, and seeing if we could map where on the spectrum someone would lie based on the particular type of dysregulation and its degree. Now, what I just showed you should be a little bit, this should shock you a little bit, because I just talked about, gating mechanisms, and yet this model, it has filters, it has comparators, it has gains, and so forth. And for those of you who are neuroscientists, you are probably asking, well, wait a second, I mean, does the brain really have, comparators and gains, and, is that real, or is that just a way to make the simulation work? So this is exactly the question that we want to be able to address. So the question really is, what are the computational primitives of brain circuits? So when we say that the brain is processing, what is doing the processing, right? Again, it's not just that this is connected to this, right? And this is connected to that. That's not going to process anything. So what is doing that processing? So in the case of an electrical circuit, we know what those computational primitives are, right? These are synthetic circuits. We have capacitors, we have gains, we have resistors, and so forth. And so the question is, even though in computational neuroscience, sometimes we use analogs for those, we'd like to know that those analogs have an actual underlying biological basis. So in Neuroblox, those are represented. So these underlying processing elements at the bottom scale are described as computational primitives, biomimetic computational primitives, because they're meant to reflect actual brain processes. And here's an example of one of them. So this is lateral inhibition. And if you see how this works, so you have a number of excitatory inputs, and the first one that hits inhibits the rest. Okay? So I just want to point out something. First, again, this is not just who's connected to whom. There's a critical timing issue. The first one that hits inhibits the rest. So any model that doesn't consider the timing element, it would be missing something critical about this function. And second, this is acting as a type of filter. And depending on how you tune it, it can have different effects, but it is one potential mechanism for a filter. So one of the tasks that we've had in Neuroblox is figuring out what are these computational primitives. These computational primitives are mostly at the spiking neuron scale, and they're really microassemblies. They're very tiny structures that have a very particular function. And the way we define them is the smallest possible function, the smallest possible structure that can perform a particular function. So here is a sample of these potential computational primitives. These are not made up out of our heads. These are coming from data, from the literature. They need to be figured out. And then from these computational primitives, we can arrange these microcircuits in ways that allow us to design macrocircuits. So in other words, you have a bunch of microcircuits that are arranged to create a macrocircuit, then that can be treated as a block that then is used to arrange a larger macrocircuit. And this is how Neuroblox grows in scale. It's circuits of circuits of circuits of circuits all the way up. Okay. So here's an example of this particular circuit, and as you can see, so this is the corticostriatal circuit, and it's doing a particular task. So you might recall that I said that one of our desiderata is we want our model to be actually capable of producing some effect. So we want it to be able to do a task. And in this case, it's going through a series of steps. So it has, this is, again, all the way, starting from the spiking neuron scale, all the way up to what will be measured with the local field potential scale. And then through that to behavior. So these different components that were assembled from the literature. So this is a kind of an integration of many, many decades worth of experiments, data, models, all got normalized into this Neuroblox type of nomenclature and then combined together to create this comprehensive corticostriatal model that can do category learning. We then independently validate it against macaque electrophysiology. Okay, now again, the only reason we can do that is that it's producing simulations, so predicted outputs as trajectories at multiple scales, spiking neuron, local field potential, and behavioral. And it's being checked and validated at multiple scales to make sure that this is actually correct. Now, this brings me to the next question, which is the user experience, the GUI. How do we interact with this? What we want is, again, to create a tool for neuroscientists, of course, but also for neuroscience to be actually extended to developing better drugs and devices. So that means that it can't be the domain of a small select group of hardcore computational neuroscientists who live in a cave somewhere. It needs to be something accessible to many different types of populations, experimentalists, theoreticians, and so forth. And so we really thought this through very carefully, again, with those aims in mind. So the tool that I used with my team to derive that fMRI scale control circuit for the prefrontal limbic circuit was done in Simulink. But Simulink, it was really developed for electrical engineering. It has computational primitives that are really specific to electrical engineering, which means that it has the advantage of being able to allow you to model in a graphical way, which allows you to tap into your intuitions, your visual intuitions about what a circuit looks like. It also allows you to work at multiple scales, so you can have blocks within your Simulink that are made up of other circuits underneath it. So it has many similarities to what we're trying to do. But again, this was optimized specifically for electrical engineering, and we want to make sure that the tool it's sort of optimized for the sorts of questions neuroscientists would want to be asking, and also, ideally, also kind of keeps you from making mistakes that Simulink would allow you to make simply because it doesn't have biological constraints built in. So one of the practical problems we ran into very quickly is that when we're looking at circuits, and if you want to map this onto a brain, which is the geography that neuroscientists are familiar with, the problem is that circuits are usually not all in one slice. So if we take one particular slice, we're going to have to be a little bit loosey-goosey about putting the blocks where they belong, because it's not, we're going to have cortical areas, subcortical areas, and front and back and side. And so we had to come up with a way of doing this that would allow the entire circuit to be visible at once. And so, kind of inspired by a Mercator projection map, we thought, okay, can we actually create a flat map representation of the entire brain? And we were able to make use of some wonderful work by Hahn and Duckworth in 2023 that did exactly that, and we were able to adapt from this. So here is the GUI that Neuroblox uses. And just to walk you through how to work your way through this GUI, so the blocks, which include computational primitives, but then it also includes the blocks that are these circuits that are created, the macro circuits that are created from these computational primitives. And so then, again, the blocks of blocks of blocks can also be included as blocks, which means that when someone creates a circuit, they are using the same blocks, okay? The advantage also, by the way, in having different groups work from the same common set of elements is that it accomplishes something that all too often doesn't happen in computational neuroscience, which is the ability to compare one model to another and to integrate models. So computational neuroscience tends to be a very artisanal activity where every group has its own ways of defining the circuits and their interactions, their interaction rules, the ODE solvers even. And so the advantage of having this common set of elements that can be combined is like in electrical engineering. If someone says resistor, I know what resistor means. I can change parameters for the resistor. I can create another type of resistor, but a resistor has a common meaning where multiple groups can combine circuits, electrical circuits, in meaningful ways. But by the way, I should mention, you can create your own blocks. It's not as if that you are stuck with the blocks that already are there. You can use your own data or other people's data to create new blocks. And then you have your blocks are arranged on the brain. So I don't know if you can see, but these are actually brain regions. Can everybody see the brain? Yeah. So this is a flat brain that includes different brain regions. And so you actually put your block on the brain region. If you select the brain region, it will automatically put the block where it belongs. And the reason why we want to put things where they belong is partially because, again, we have, as neuroscientists, certain intuitions about the brain, right? If we see something in the visual cortex, we know what that means. If it's in the striatum, we know what that means. And so we want to have things where they belong. But there are also other important advantages of having there be a spatial geography that is meaningful. And one of them is that you can actually have mechanistic maps that overlay with this map, this brain. And so if you have, for example, you have different, GLUT4 density distributions throughout the brain. If you know what the GLUT4 density distribution is throughout the brain, you can map it onto here, and that will change the underlying parameters of how your model is running. The last thing I just wanted to point out is these connections. So this is arranged on the left between inputs and outputs. So the inputs can be drugs, devices, stimuli, anything that is actually going to go into the system. And then you have what comes out of it. And so what comes out of it could be various ways of visualizing your data, but also motor action, for example. So if the simulation is conducting a particular task in response to some particular type of stimulus, the output would be the behavioral output. So this is being adapted to a couple of different applications. One of these is a pharma module. So one of the things we want to be able to do is to ask, if we have different drugs, how are they affecting not only neurotransmitter levels, but also receptor dynamics, right? So we need to be able to model receptor dynamics. It means that when you tag a particular connection, you will identify what neurotransmitter that is, and then you have the opportunity to include relevant receptor dynamics. Obviously, this at this level is going to affect the computational primitives, which means that then those computational primitives will affect the running of the circuit as a whole. And so when you ask, what is the impact of giving this drug versus that drug at this dose versus that dose, it's affecting this level, right, which is then going to be affecting the computational primitives. And then the computational primitives are going to have emergent effects all the way up to the behavioral scale. We also have a metabolic module. This is composed of metabolic flux equations that, for those of you who do magnetic resonance spectroscopy, allows you to ask, how will changing metabolism of this parameter versus that parameter, how will it change the behavior of the system as a whole? Even at the level of changing someone's diet, putting someone in ketosis versus not. Giving someone type 2 diabetes. How is that going to affect the entire, so this will affect the system, the metabolic system, but then this outputs neurotransmitters. Again, those neurotransmitters are going to go into that circuit, and then the circuit will run its simulations and have some output. And that allows us to ask a whole new category of questions like, why is it that GLP-1 agonists seem to curb addiction? Well, in order to answer that question, we're going to need to be able to model the metabolic system and also ask how it impacts the neural circuits, right, including behavior. And so one of the things that we're very excited about is the ability to not only model circuits within the brain, but also the brain's interactions with other systems, for example, the immune system. If you've ever thought about long COVID and its impact on brain fog, again, these are really important clinical questions to ask. And then we have also a neuromodulation module in progress, where we're interested in being able to stimulate the circuit in various ways with DBS or transcranial magnetic stimulation, and ask, okay, so how is this affecting the system as a whole and its behavior over time? So finally, I want to say that kind of one of the grand goals is to develop Neuroblox as a knowledge integration ecosystem in a way that I don't see existing platforms have kind of quite gotten us there yet. And one of them really has to do with allowing for the ability of community-based modeling, where if one team is contributing data and another team is contributing models and another team is contributing other models, that there can be an integration and comparison of the models to the data, the models to each other, in a way, as I said, that's only possible if you're working from a common set of modular elements. All of this gets powered by Julia and facilitated this hosting for this week, but probably also long-term will be through Julia Hub. Obviously, this is very computationally expensive, and so the capabilities that Julia offers in terms of high-performance computing are really critical. So Neuroblox allows for simulations and model fitting. The modularity that I've described is designed from the ground up for automating a lot of the processes that otherwise wouldn't be possible. So creating models, testing models, fine-tuning models, parameter fitting, comparison of models, et cetera. Global parameter setting, so again, putting together a whole bunch of different models, asking how they integrate, and then being able to change parameters for the whole system from one place, and model comparison and integration. And now Helmut Strey, who's our lead software engineer, will take over.
So I wanted to spend a few more minutes on the more technical aspects of Neuroblox. In particular, we want to talk about why we chose Julia over other software platforms. And to start, I wanted to introduce what is coined the two-language problem in computational science. And that has to do with, if you want to do something performant, right, you want to have a low-level programming language that is really fast, right? And that's for execution speed, right? Because you don't really want to have a slow simulation, right? Because typically you want to push the envelope. But on the other hand, if you have a lower level language, it's very hard to develop code, and to increase development speed, you need a higher level language, right? And so typically what people do is, and this is even true in computational neuroscience, is that typically they use C or C++ for really performant code, and then maybe use Python to interface with that C code, right? So there's libraries that essentially say, build me a neuron assembly, and then that gets translated into C. So the nice thing is that Julia actually solves these two language problems, because it is actually the motto of Julia is Julia walks like Python and runs like C. It's a compiled language. And it actually, if you look at performance, there are online Julia performance benchmarks. It's very much like C. Sometimes actually we beat C in performance because it's better optimized. So that's the first thing, right? That was our first decision. The other decision is on performance and accuracy of simulation of differential equations. Julia, I think, has, and I think it's pretty, it's a true statement that Julia probably has the best performant differential equation solvers in the whole field, and not only ODE solvers but also SDE solvers and delayed differential equation solvers, and actually also differential algebraic equation solvers. And here are some benchmarks that I want to show you. So these are non-stiff solvers and stiff solvers. And one of the things that you will see immediately is that Julia performs about 2 orders of magnitude faster than Python or MATLAB, right? Which is not super surprising because MATLAB and Python are not very high-performant languages. And they perform very almost equal to any implementation that you can do in C. So this was one of the reasons. I don't think there's any other language that has so many choices in solvers, and that's one of our motivations. That when people use the software, they can actually pick the right solvers, right? And this is why I'm actually showing non-stiff versus stiff. Non-stiff are differential equations where the functions are relatively smooth and nice and well behaved, and then you can essentially work with adaptive solvers. Stiff solvers are more for hard problems like chaotic systems, things that are oscillating, for example, highly nonlinear systems, where you have to have different strategies to solve differential equations. And you can actually read this up on the benchmarks here, down here. The other thing that we were very keen about is differentiability. Julia has a large automated differentiation ecosystem. And I don't know how many people have heard of auto differentiation. Some of the, right? So auto differentiation, I mean, you probably clearly heard for machine learning and neural networks. They call this backpropagation. Backpropagation is essentially reverse auto differentiation. It's the same. It's a different word. In different fields, you use different words, but it's essentially algorithmic, the same thing. The reason why I think it's important to say automatic differentiation is because there's also forward automatic differentiation, which you use when you only have about 100 parameters. Then actually forward differentiation is better than reverse differentiation. Reverse auto differentiation is really effective when you have a lot of parameters. So for example, in neural networks, you have 10,000s, or the big models have like billions of parameters. Then backpropagation is really the method to do. And we have all these libraries essentially in Julia where you can apply auto differentiation to any function in Julia. I mean, maybe not any function, but any function that it's like in math, right? Like what is integrable and what is differentiable, sort of like these kinds of things. And the reason why we're so excited about auto differentiable is because then you can address problems that you couldn't before address, right? One of the things is, what do you need differentiability for? Optimization, right? So, for example, if you have a wonderful model, right, that you just discovered and vetted and it's the best model that you can come up with, right? What you would like to do is to then take data, patient data, right, and then fit parameters to that particular model, right? And you can only do this if you actually are good at differentiation, right? So optimization problem, the inverse problem, that's the parameter fitting problem. Then there's other applications that we actually pursuing in Neuroblox, and one is optimization of the system itself, right? So let's say you have a collection of neurons. You pick the neurons because they're the ones that you want to use, but you need to optimize them in terms of performance. You want the right firing rate, you want the right frequency spectrum, and stuff like this. And so what we can do is we can essentially put constraints on the performance of these collection of neurons, and then do an optimization process through optimization.jl. And the other thing that you can think of is then if you go the Bayesian route, the Bayesian route would mean that we don't necessarily do parameter fitting. We don't want to fit the best parameters. Well, we would like to find the posterior probability of the parameters given the data, right? Then you would go in the Bayesian route. And with the Bayesian route, there's a lot of packages that allow you to do a Hamilton Monte Carlo on probability distributions, right? So that would give you the full posterior probability of your data. So the other reason why we picked Julia is because of composability. So Julia has in part of the suite of SciML tools, scientific machine learning tools, has a package that's called Modeling Toolkit, which uses symbolic equations to form differential equation systems, ODE systems. And I just want to give you a quick example. So these are actually two blocks in Neuroblox. Each block is defined by a differential equation. And you see there's a very small junction in there. Within the sigmoid function, there's a little junction in here. And what you can do is you can take, for example, the output of one block and connect it to the junction of the other one. And then you can connect this X to the junction of the other one. And I indicated this by these arrows. And that's exactly actually how you will see this in the GUI. Every arrow essentially means that I connect an output of one of the ODE systems to an input of the other ODE systems. And so what Modeling Toolkit essentially does is it takes all these equations and then cleans them up. It, for example, removes the junctions, right? It eliminates the junctions because the junction is a connection, right? And then turns that into a function that then can be solved with an ODE solver. So this is full composability. And so the way we constructed or designed Neuroblox is by using a hierarchical and composable structure, right? So for example, you start with a Hodgkin's-Huxley neuron, you put 5 of those or 6 of those neurons together into what we call the winner-takes-all circuit. Actually, Lily was talking about that exact circuit. And then you can take all these winner-takes-all circuits and put them together and make, for example, a cortical region, right? And assemble them. Now, the nice thing about this hierarchical structure is that you also have hierarchical namespace. So if I have a cortical region, I can ask, so give me the first winner-takes-all circuit within the cortical region, and then give me the second excitatory neuron in that. And that's essentially a namespace that you can walk through. And I think in the tutorials, we'll see exactly how to use this. We can also have shared parameters. So you could, for example, say, we'll do a cortical region, and all the parameters of all the neurons are the same, and they are one number, right? And then you can tune that number rather than having to tune 5,000 parameters at once. We can also actually define observables, so we can put in functions that then can be later, after the simulation, be extracted from the system. And actually, this is something that Harris developed. We have connection rules. So if you have blocks and you have other blocks, then you can essentially create rules how to connect one block to another block. And it's very flexible. And you will actually learn during the tutorial, I think, to make your own blocks and your own connectors. It's going to be quite fun, actually. I want to talk a little bit about Neuroblox performance. When we first started this project, we noticed that because we start with symbolic equations, it actually gets really slow. So when you build big systems, there's a lot of simplifications that has to be done. And Modeling Toolkit was actually not up to the task to give us fast compilation. So what we noticed actually that when we have really large systems, fully connected systems, then the compilation time is much larger than the execution time. And that's really bad. So actually, Mason was the driving force. He developed a new way of creating ODE systems where we take advantage of the fact that we have the same thing over and over, right? So we have a neuron, for example. The neuron rules of connections are always the same. And then you can essentially take advantage of that structure. Actually, what we take advantage of is that every system that we create is actually a graph, right? Because you have nodes, you have edges, and the edges define the connections. And creating equations out of graphs is much more effective. And so we created actually a library, Graph Dynamics, that compiles graph systems much more efficiently. And you can see modeling toolkit is on this is the blue graph, blue data, and Graph Dynamics is red. And so we are, again, orders of magnitude faster than modeling toolkit. I'm going to conclude by saying that we essentially have the following features. We are composable, hierarchical. We can build neuroscience models from computational primitives. We have a native high-performance computing language. I mean, Julia, by the way, I didn't say this. Julia is about 10 years old. It was developed here at MIT in the math and computer science department. And it's actually a language from mathematicians for mathematicians. But engineers can also use it. It's I think, fine. But it's designed for high-performance computing. So interface with GPUs and multi-core CPUs. By the way, Julia Hub is a way of running Julia in the cloud, and you can throw essentially any computing power on your problem. So we have differential models for optimization, parameter fitting. That also then allows us to do computational model comparisons, right? Because if you have two models, they're all differentiable, and we can do parameter fitting, then you can essentially do Bayesian posterior ratios, and then you can essentially tell which model is better. And that's actually one of our plans in Neuroblox, that we, could essentially have people, submit different models, and then we can rank them. We can say like, okay, so how well do they do with respect to the data that is available? And so we also will have a graphic user interface, which I will present right after this. So thank you so much.